All right, let's dive into something pretty mind-blowing. We're talking about the science of growing entire plants, not from seeds, but from just a tiny, tiny piece of a plant, all inside a lab.
Okay, so let me just throw this out there. Could you grow a whole forest inside a single test tube? I mean, it sounds like pure science fiction, doesn't it? A whole world in a little piece of glass. But what if I told you the science to do exactly that is not only real, but it's been in the works for over 100 years?
The secret sauce, the magic word here, is totipotency. And honestly, it's one of the coolest things about plants. See, unlike most animal cells, a single plant cell, yeah, just one cell from a leaf or a root, has this superpower. It holds the entire blueprint to grow into a whole new plant. A perfect clone. So, knowing it's possible is one thing, but how do we actually figure out how to do it? Well, that's a story that took decades of really patient work, figuring out the keys to unlock that hidden power. It all kicked off as just an idea way back in 1902. Then, fast forward to 1939, and scientists finally managed to grow these weird blobs of cells called calluses. But the real game changer? That was 1957. Skoog and Miller totally cracked the code. They figured out the exact hormone recipe that tells the cells what to do. And then boom, by 1971, they were growing a whole plant from a single cell that had its wall stripped away. Just incredible progress.
Oh, okay, so how does this actually work in the lab? You can't just, toss a cell in a jar and cross your fingers. No way. You need a super specific recipe and an environment that is unbelievably clean. We're talking cleaner than an operating room. It's basically a 5-star all-inclusive resort for plant cells. And the first rule, the absolute non-negotiable #1 rule, is that everything has to be perfectly sterile. I mean it. One little stray microbe, a single fungus spore, and your whole project is toast. That's why scientists do all this work inside what's called a laminar flow hood. It's constantly blowing a curtain of super filtered air to keep any nasty contaminants out. Okay, so once you've got that perfectly clean workspace, the real process starts. First, you take a tiny piece of the plant, they call it an explant, and pop it into a sterile jar. Then you set it on this special nutrient gel that's like a buffet of everything a growing plant cell could ever want. From there, it goes into a growth chamber with the perfect lighting and temperature. And then, once you've got a little baby plant, you have to slowly, carefully get it used to the real world.
And just like people, not all plants want the same nutrient smoothie. Picking the right food is super important. The most common one is called MS medium. It's kind of the all-purpose, nutrient-packed choice for most things. But if you're growing cells floating around in a liquid, you'd probably go for something like Gamborg's B5. Or if you're just trying to get roots to grow, a low-salt option like White's medium is the way to go. But here's where it gets really cool. Scientists aren't just watching this happen, they're in the driver's seat. They can actually steer the growth, telling the cells exactly what to do using chemical signals. Think of it like a control panel for the plant. And that panel really has two main knobs. There are two types of plant hormones. One's called auxin, the other is cytokinin. And getting the balance between those two just right, that's the key to everything. It's this amazing biological tug of war. If you crank up the auxin, you're basically shouting at the cells, grow roots. But if you dial back the auxin and turn up the cytokinin, the message completely flips to grow shoots. And the results are so predictable. A little more auxin, you get roots. A little more cytokinin, you get shoots and leaves. And what if you get the ratio perfectly balanced, right in the middle? Well, the cells just kind of sit there and multiply without becoming anything specific. That's how you get more of that callus stuff. It's just a stunning amount of control. Okay, so we have all this incredible power. What do we do with it? Because this is way more than just a cool lab trick. This technology is literally rewriting the rules for everything from farming to medicine. Check this out. It's called somatic hybridization. Just think of it as creating a plant mashup that nature could never make on its own. Scientists can literally take cells from two completely different plants, strip off their cell walls, and fuse them together. That new hybrid cell can then grow into a whole plant that has traits from both parents. It's how you can take, say, disease resistance from a tough wild plant and give it to a fragile, high-yielding crop. But the possibilities just keep getting wilder. I mean, what if a plant cell could be a factory, a tiny, microscopic factory for making medicine? Well, that's exactly what's happening with something called plant cell bioreactors. Scientists grow these cells by the vatful in these big liquid tanks. And get this, they can add elicitors, which are basically just stress signals, to trick the cells into pumping out massive amounts of valuable stuff. This is how we can get things like the anti-cancer drug Taxol from yew tree cells. Or how about the flavor of real vanilla without ever touching a vanilla bean? It's all done in a lab. Beyond making new things, this tech also offers some amazing ways to preserve what we already have. It's basically moving conservation out of the field and into the lab. So think about this for a second. How do you back up the world's most important plants? Or the most endangered ones? I mean, seeds are good, right? But some plants barely make any, and seeds don't last forever. We need a better way, like a way to hit pause and put them in suspended animation.
The answer is cold. Really cold. We're talking negative 196 degrees Celsius. That is the insane temperature of liquid nitrogen.
The technique is called cryopreservation, and what they do is take tiny bits of a plant, or even just cells, and carefully freeze them solid in liquid nitrogen. At that temperature, time basically stops for them. All biological activity just ceases. It is the ultimate backup plan, a deep freeze that can save the genetics of our most important crops or pull an endangered species back from the brink of extinction centuries from now. So there you have it. From the secret power hidden inside one single cell, we've figured out how to create totally new plants, brew medicine in giant tanks, and protect the planet's biodiversity for generations. It really makes you think, if the lab is becoming the new farm, what on earth are we going to grow next?